Today I'm going to be focusing on variation, which is in your Unit 4. So I'm going to start with a few questions for you. Possibly you've done this topic already, but you also will have done it at GCSE. So I just want you to think about, what is the meaning of the word variation? What does it mean? What variation can you think of within organisms in your own home? So I'm not trying to give you too much detail there. How can genetic diversity be increased? How can we increase the amount of diversity within our genes? And how can it also be decreased? I'm going to give you a moment to think of those questions. So hopefully you know the meaning of the word variation means different. So what differences can you find within organisms in your own home? So for example, just within a classroom setting, you might notice things like hair colour, eye colour, etc. Okay, the nose shape, lip shape, ear shape, and so on that you did when you studied this at GCSE. How can genetic diversity be increased and how can it be decreased is something we're going to look at in this session. So we're going to look at variation, competition, population genetics, and Hardy-Weinberg principle. So differences in phenotypes, so variation in phenotypes of organisms. So we need to remember that phenotypes are the characteristics that show, they're the colour that shows, for example. So we're not talking about the genotypes yet, which would be, for example, little b for blue eyes. We're talking about the phenotype, which is blue eyes. So why do these differences occur within organisms? So first we have the different genotype. That is going to be one of the reasons that we have the recessive, the dominant, or we have a recessive and dominant together. So we have our heterozygous and our homozygous genotype. Then I'm going to come here next, different environments. So organisms tend to live in different environments to one another, and that can also affect our phenotypes. It can affect things that are quite visible. So at GCSE, you would have looked at things like the length of hair or the style of hair. And then we have this one in the middle, which is the same genotype, but different epigenetic modifications. So this means that the genes are expressed in a different way. So epigenetics are a study of how your behaviors and environment can affect the way your genes work. So they don't change your DNA sequence, they just change the way that your body reads the DNA and therefore affect the way your genes work. So hopefully that will help you with understanding what this means. Okay, if you need to, obviously when you watch this as a recording, you can rewind. So let's look at heritable variation. Now they've used this term at A-level, heritable, but it's the same thing as inherited. So it's something that you get. So if there are different DNA nucleotide sequences or different epigenetic modifications, they can be inherited. So I can inherit the DNA nucleotide, the genotypes, and I can inherit markers that tell my genes to work differently. In asexually reproduced organisms, heritable variation is only increased by mutation. They are clones, so they're going to be genetically identical unless a mutation has happened, and at that point we'll have some variation. In sexually reproduced organisms, there are many mechanisms that bring about variation. I want you to try and have a think before I change the slide. What brings about variation within, you can think of humans, what brings about variation within sexually reproduced organisms, such as the humans? So it's a bit of revision of other topics, to be honest. So sexual reproduction causes variation due to the mixing of two different parental genotypes at fertilization. We've got half the information coming from the sperm and half coming from the ovum, therefore mixing parental genotypes. Then we have meiosis revision. Okay, so again, it might prompt you to have a look over this. So we have the crossing over between homologous chromosomes. You might remember that they line up and there's crossing over between the chromosomes in prophase 1 of meiosis. Then there's the random distribution of chromosomes during metaphase of meiosis, metaphase 1, sorry. And then there's the random distribution of chromatids during metaphase 2 of meiosis. And all of those bring about variation within sexually reproduced organisms. Non-heritable is that it can't be inherited. Okay, so they've used new terminology, but it means the same. So this is where variation has happened due to the environment and not a result of epigenetic change. So it's not happened because of your environment affecting the markers. So this has happened due to other things. Okay, so preferences for certain types of food. Okay, that can be due to the environment, but it's not going to be passed down. It's not going to be inherited. So mutation itself. So the processes we have stated already create new combinations of alleles. So these four here create new combinations of alleles in each generation. But it's through rare mutations that long-lasting variation happens. And I've put more detail down here, and I'm just going to read this for you. So most mutations are recessive to the normal allele. They're not going to change the dominant allele within a species. So the recessive mutant allele can only, remember, be expressed, be shown, the phenotype can only be seen if there are 2 copies of it. So that would mean both needing to change, which is obviously infrequent. So it has to wait for replication in the gene pool. So it's there, it exists, but it has to wait for another one the same to be there. Okay, so it takes lots and lots of generations usually before that chance brings the 2 recessive alleles together, which then results in us seeing the phenotype. We've looked at heritable and non-heritable. We now need to look at the 2 types of variation. So again, you will have studied this at GCSE, but there's a little bit more detail. So discontinuous is where there are groups. So for some characteristics, the number of possibilities are quite small. So you would have studied Gregor Mendel's plants at GCSE most likely, and he looked at tall and short plants, what he called dwarf plants. He looked at wrinkle peas seeds and smooth pea seeds. He looked at purple flowers and white flowers, etc. So the possibilities, the number of possibilities is quite small. It's this or this. The other thing about discontinuous variation is there are no intermediate types. What this means is they're groups. You don't have a mixture. And what we call them is, what WJC calls this is discrete, which actually means easy to tell apart. Okay, so that the use of that word can be a little confusing. What it means is that it's easy to tell them apart. So I know it's easy to tell apart brown eyes from blue eyes, for example. They're generally, as you can see here, controlled by single genes, which means they are mono meaning one, monogenic. The environment has no influence on the gene expression. It has no influence on the phenotype being shown. Okay, so that's your discontinuous. It's groups. They're usually not many possibilities, and it's easy to tell the difference, controlled by one gene, and the environment has no influence at all. And then we go to our continuous variation, which is where you see a sort of range of results. Okay, so I've written here, there can be many possible values. So human height between both extremes, you'll see on my second point, which is the shortest and the tallest. We could refer to those as the extremes, the furthest away from what we would consider the norm. You'll see in a moment we talk about mean mode, etc. So the values show a range of possibilities from one end of the spectrum to the other. Okay, I can't group these people. I can have heights all along. Okay, weight can be another one. It's controlled, or they're generally controlled by many genes, poly meaning many. So continuous variation is generally because there are many genes involved. And the environment does have an influence, or can have, okay, at least. So it does have an influence on gene expression and can determine the phenotype variation. So we'll have a look at that in a bit more detail now. So human height is an example of continuous variation, and alleles that determine potential maximum height. Okay, you do have alleles that determine how tall you will be, but the amount of nutrition you receive, the amount of food, and the amount of exercise you do can affect it. So that's your environment influencing how tall you will actually be. Okay, so a simple example is if I am a malnourished child, then I probably won't reach my full potential in terms of height. Okay, and we can look at number of leaves a plant has. There are different factors as to where the plant is, how much light it gets, does it have all its mineral requirements, as to how well the plant grows and how many leaves it has. So I want you to think now, which type of graph is usually used to represent data for continuous and discontinuous data? I don't really need you to think about variation at this point. Just how would we show data that has continuous range of results? And how would you show data in a graph for discontinuous data? Have a think for me. And I have put a big hint on the screen. Okay, so if we are thinking back to earlier times in science, then continuous and discontinuous data without thinking about variation, hopefully you've thought, well, if there are groups, so I would usually say names versus numbers or words versus numbers, we have a form of bar chart. So that's going to be your discontinuous. Your continuous, okay, so if we're not thinking about variation, continuous means that we have a set of results, okay, either one of these. So this is where you would plot little x's and you would have your line of best fit. So it's the same thing. And here we go. So continuous variation, you can see here from both extremes. So here's one extreme and here's the other extreme. So the lowest it can be and the highest it can be. We have a range of results shown here by this curved line. And what we see is that there's normal distribution around the mean. So the mean, okay, we'll go into more detail in a second, the average, okay, there's going to be a lot of people at around average height, and it'll scale off to the most extreme ends. Okay, there aren't that many people who are 6'7", for example. Okay, so they will be up here and there will be fewer. Okay, and you can see here median and mode are the same. I've got another slide to help you with this. Discontinuous variation is dwarf and tall pea plants. So we'll have two sets of results here. So there will still be some variation. Okay, we're still going to see some variation within the height of the plant, but we've definitely got two distinct groups here. Here, I can't tell apart the groups in that curve. So let's look at these keywords in more detail, and I've added some information below. So your mean, as you can see here, is your average value. Hopefully you're well accustomed now with calculating the average. You add up all the numbers and you divide by the number of values you have. Okay, so there's an example given below. I add them all up. I've got 4 sets of results, so I divide by 4. The median is the middle number. So you put the numbers from smallest to largest, and the number in the middle is the median. Okay, so you can put them all there, divide by 2. okay, which just says here, add them up, divide by 2. Mode is the most frequent number, okay? Thinking about everything we've gone through already, I want you now to compare continuous and discontinuous variation. In an exam, if they ask you to compare or distinguish between something, you are allowed to split your lines into 2 and put continuous and discontinuous. What you need to make sure that you do is you comment on the same thing on both sides of the table. Or you can write in paragraphs, but I'd recommend that you talk about both at the same time rather than leaving one to the end. You often lose marks if you don't directly compare. So what I mean is you say, this occurs in continuous but does not in discontinuous. Continuous does not have this, but discontinuous does. Rather than saying everything for continuous and then underneath everything for discontinuous. You don't want to lose marks. So you can do the same here. So what I'd like you to do now is compare continuous and discontinuous variation. I'm going to wait a few seconds and then I'm going to put up a little bit of help in case you're a little bit lost. I'll let you know when I'm going to do that so that you can pause the video if you don't want any additional help. Okay, I'm going to put up some additional help to help get you thinking. So now that you can directly compare them, does continuous have it? Does continuous have it? Okay, this isn't how you'll answer your exam, but you can use this right now to help you start comparing. So is it heritable? Tick or cross. Is it non-heritable? Tick, cross. Okay, and I'm going to put the answers up, so if you need to, you can pause the video. So I've given it to you like this, okay? So you can look at the relevant comparison. So no distinct categories versus distinct categories. No limit on the value. There are no in-between categories. It's almost saying the same. Height, weight, heart rate, for example, are examples. And over here, you might remember back to cystic fibrosis, fingerprints, blood groups. Again, it's represented by a line graph, and this one is represented by a bar graph. This one is controlled by many genes, okay, and one or very few genes. This one can be affected by environment. This one is not. Range of phenotypes between 2 extremes. There are no intermediates. There are no groups. Okay, and you can write examples to show that. Okay, here is an exam question from the 2018 paper, which you can find online. Just going to give you a moment to read that information before I switch the slides and you get to see the question. Okay, I'm just going to give you a slight tip on this. If you're still reading, you can pause the video. Now, this text here isn't that relevant for the question we're about to answer, but there is a reason they put this into the exam. And I know it's quite common for students to skip this bit and just go, yeah, there's nothing relevant, I don't need that, and go to the question. But it is really important you actually read this information. I have only put part of this question in this evening because it links to variation, but the other questions are linked more to looking at this text. So here is the graph. I'll give you a couple of moments just to analyse and decipher what's being shown before showing you the question. Okay, and here is the question. And I've got a small version of the graph at the bottom. Hopefully it's big enough for you to see. So you need to state the type of variation shown by the plants taken from the unpolluted soil. Explain your answers. And with reference to the graph, state which letter, A, B, or C, identifies the mode for sweet vernal grass taken from the mine site, and state the difference between the terms mean value and modal value. So you've got to state the letter, but you've also got to give the difference between two different terms. Have a go at those questions. Okay, I'm going to move on. So again, you can pause the video if you need to. So it's showing continuous because there aren't separate groups. Hopefully you got that. And here it's B, okay, so you can see there it's B. And mean is the average of the group, and mode is the most frequent, the most common value. Let's look at competition quickly. So I've written lots on this slide to help you. So when there's no environmental resistance, organisms overproduce. So their generations get bigger and bigger from one to the next. However, that's really uncommon in a natural environment, and there are usually limits to population growth. So resources can be limited, which forces competition. There can also be predator-prey relationships, which obviously brings about its own sort of competition. Therefore, because there are limits to population growth, because of resources being limited, there are fewer offspring produced, or the offspring themselves might not make it to reproduction age. They might die before maturity, so they don't produce, therefore it's not increasing the generation that's coming. You need to know about two types of competition. Now, you need to remember that intra-specific competition happens between individuals of the same species. So same animal, same habitat, for example. Inter-specific competition is between different individuals of different species. So for example, zebra and an antelope have inter-specific competition because they have a similar diet. So there will be competition between them to get food. Okay, so you're going to have to find a way of remembering intra and inter-specific competition. So what type of competition would occur between the flamingos and for what? Have a think. Hopefully you can say that it's intra-specific and they would compete for something like food. So here is another question for you to have a look at. Okay, I'm going to put the answer up, so if you're not quite ready, again, you can pause the video. So inter-specific, hopefully you can see why. And you could say that they are competing for space, light, water, minerals. You can give specific examples or any specific nutrients. Can't accept carbon dioxide. So we're going to move on to looking at the gene pool. So the gene pool means all of the alleles of all of the genes of all of the individuals in a population at a given time. So it's looking at every individual, looking at their genes, and looking at every allele that they have. So all the alleles present in a population at a given time is the gene pool. Let's look at some key terms that we need before we move on to some maths. So allele frequencies are the genes and alleles of the entire population. Genetic diversity means the genetic differences between the individuals within a population in terms of alleles. So how different are their alleles in this population of one type of thing? The genotypes of the individuals are a selection of the allele combinations that are possible at the time. So for example, big B, big B, big B, little b, little b, little b. So it's the genotypes of the individuals that exist at that time. How often a particular allele occurs within a population is called the allele frequency. So how frequently we see the allele. So the frequency of an allele is its proportion, fraction, or percentage of all the alleles in that gene pool. How often do we see it? So what's the proportion compared to the whole gene pool? What's the fraction compared to the whole gene pool? Or what's the percentage compared to the whole gene pool? So we need to look at the Hardy-Weinberg principle. This is considering in ideal conditions. So in ideal conditions, the frequency of dominant and recessive alleles and genotypes remain constant from one generation to the next because nothing's forcing them to change. So we're looking at ideal conditions here and that everything stays the same, remains constant. And these are the ideal conditions. So first of all, we need the organisms to be diploid with a single gene and two alleles controlling the gene. So we're just talking about a specific gene here, but it's a single gene that controls it. Nothing with multiple genes. We're not going to be looking at human height. We need the allele frequency to be equal in both sexes, in male and female, in both genders. Reproduce sexually, okay? We need mating to be random, which of course would happen within the natural environment. And we need the generations not to overlap. Okay, so you can see these ideal conditions aren't always available, but this is what we're basing this on. The population size needs to be very large. There needs to be no migration, no mutation, and no selection, okay? So these are the ideal conditions that we're looking at for Hardy-Weinberg principle. So let's look at the fact that there'll be no evolution. So the Hardy-Weinberg principle also shows that if these conditions remain constant, no evolution will happen because nothing's going to change. So there's going to be no change in the frequencies of dominant and recessive alleles, and there are no selection pressures to favor one phenotype over the other. There's nothing that's going on that would favor the color of a moth, for example. So if you assume that these conditions are met in a particular population, the equation can be used. So we use these equations to calculate the allele and genotype frequencies in a population and to predict the number of individuals in a population carrying that certain allele. So these are the equations that we use. So you need to know that the frequency of alleles, we look at P plus Q equals 1. And the genotypes, we look at P squared plus 2PQ plus Q squared equals 1. So if P refers to the dominant allele and the Q refers to the recessive allele, can you suggest what the terms in the second equation represent? Why do we have P squared, Q squared, and why do we have 2 times P times Q? Okay, I'm going to go on to the answer. So P squared is the proportion of a population who are homozygous dominant. So we're looking at AA. So P itself would have been A, but P squared is obviously what the genotype will show potentially. So we need to look at A, capital A, capital A. The same can be said of the Q squared. We're looking at the recessive. So we need to look at the allele, which would be Q equals A, but we need to look at the genotype. And the 2PQ is 2 times P times Q, meaning that we look at the recessive and dominant in heterozygous genotypes. So the homozygous dominant and the heterozygotes, okay, have the same phenotypes. You can't use them directly to calculate the frequency or proportion of the dominant allele. However, the homozygous recessive only has one genotype. So what this is saying is that this and this, okay, have the same phenotype. So you can't use them directly because if you remember, if I change that letter to that for a second, you might remember your brown and blue eyes more easily. This is going to be brown eyes and this is going to be brown eyes. So we can't look at that to calculate the frequency. But we can look at the recessive because it only has one genotype. So we start these calculations most of the time with the square root of Q squared. So let's have a look. Let's try this question. So cystic fibrosis is an autosomal recessive disease. So if you have it, you have homozygous recessive alleles. Using the alleles big F and little f, or capital F and lowercase f, to represent the dominant and recessive alleles respectively, try and complete this table now. If you can't, I'm going to go through it in a few seconds. Just to give you a little bit of help, because you may have forgotten, the P refers to the dominant allele. So the frequency of the dominant allele, and what's the genotype that we're going to use. I'm going to go through the answers, so if you want to, just pause. So the dominant allele is going to be the capital F. The Q, the recessive allele, is going to be the lowercase f. I very much apologise, I haven't done subscript there. So I'm going to look at capital F, and little f, little f. Here we're going to have capital F, little f. Okay, so we now know what we're going to look at when we see these terms. So in a given population of humans, 1 in 2,000 suffer from the disease cystic fibrosis, which is caused by a recessive allele. It is caused, remember, by this. How many of this population are asymptomatic carriers? So let's go through this one together. Okay, start by writing down which term in the Hardy-Weinberg equations gets you the result that you need. So in this question, you need asymptomatic carriers. You need the heterozygotes, which is going to be the 2PQ. So use the figures in the question to calculate the frequency of one of the alleles, P or Q. So we're going to look at Q squared, which is 1. Take the number of the population that have it out of the number of the population. And this equals 0.0005. We then square root, okay? You'll remember at the start we had the square root of this, which I've calculated already. So we're going to get 2PQ. So using the formula, we have to do P plus Q equals 1. So Q squared, we've already looked at, okay? And we've got the square root here, which is 0.022. Okay, you can do this all on calculator. Okay, I did it earlier. So we'll get rid of this for a moment. Okay, so we've done 1 out of 2,000 have the heterozygous asymptomatic carrier genotype, and we square root the 0.0005 to get 0.022. P equals 1 minus Q. So 1 minus 0.022 is going to give 0.978. So now we need to look at 2PQ, and I haven't left myself much space here with this pen. So 2PQ is 2 times 0.978 times 0.022, which gives us the 0.043. We now have 2PQ. Okay, obviously, if you're watching this later, you can rewind and go through that again. And I have the working out here, okay, nice and neatly. So 1 out of the 2,000, we have the answer. We square root that answer to get what we'll need for 2PQ. We need to remember that it's the P and the Q are going to result in 1. So in order to get 1, we will take away, so we've already done this section. We'll do 1 minus Q, which we have the answer for, which will leave 0.978. 2PQ therefore is 2 times this result equals 0.043. We have now got 2PQ. At this stage of the question, you'll have to do one of 3 things. Okay, it might be that you get to stay there, but if the question asks for a frequency, leave your answer as it is. Okay, so that's the first option, is that if they've asked for frequency, leave it as it is. It's going to be 0.043 in this case. Okay, if the question asks for a percentage, you multiply your answer 0.043 by 100. And if the question asks for a number in the population, multiply your answer by the population size, which was 2,000. Here's another example that I would like you to try now. So I'd like you to try this one, and then I'll go through the answer with you. I will put some hints up as we go, so you can always pause if I start giving you things that you don't want yet because you're trying. Okay, I'm going to give a few little hints, so just press pause if you don't want to hear. We're looking at a population of 60 mice. 45 have yellow fur. Okay, so by 2 alleles, Y, capital Y for yellow fur is dominant over little y, which codes for grey. So the use of Hardy-Weinberg principle to calculate the frequency of heterozygotes, we're going to be looking at capital Y, little y. Very similar, therefore, to the last one that we did. Remember that we're going to start with Q2. Okay. So how many? So you're going to have to do a minus first. That helps. Because we're going to look at what's left from the population. Okay. I'm going to give another little hint. So again, pause me if you don't want to hear this. I'm not going to tell you what to do yet, but remember then that we are looking at the square root of that. And again, another little hint. Remember, after this point, we are looking at P equals 1 minus this answer, because we know that Q and P equal 1. I'll give you another few seconds just to try. Okay, and I'm going to talk you through it. So if you haven't quite understood it, don't worry. Okay, you can pause the video if you need to. So, yellow fur has the genotypes capital Y, and Y little y, which is what I put on the other slide for you. So the other 15, hopefully you were able to do that, but they did give you it in the question. Okay, so you didn't have to. So we knew that 45 had the other alleles. So we begin the question with this. So calculate the frequency of the homozygous recessive genotype. Let me just check, I left some space here. So Q squared would be 15 divided by the 60 that we had, which gives us the result of 0.25. Q would then be the square root of 0.25, which is going to give us the result of 0.5. You can probably see me looking at my answers from earlier. P then is 1 minus the result, which is going to leave us in this case with 0.5. So now we need 2PQ. Remember, we've got heterozygous, which means we're looking for the differences. So if we see this, we know that it's going to be homozygous. If we see this, it's going to be homozygous. We're looking for the heterozygous here. So 2 times 0.5 times 0.5 equals, anyone guessed it? 0.5. Okay. So the Hardy-Weinberg principle states that allele frequencies in a population remain constant from generation to generation, providing certain conditions exist. State 3 of the conditions. So this was the ideal conditions that needed to exist. You've just seen a flash of the answer. Okay, I'm going to show the answer, so just pause if you don't want to see it yet. So random mating, no mutations, large population, no immigration, no emigration, population is isolated. Now with these, you can only say one of them. If you've said this, you can't also get a mark for that or that, okay? And there's no selection or advantage. Okay, this is why I was kind of skipping ahead just now. If you type into Google WJC blended learning, there are a whole host, before that step, you then need to click on A-level biology, and there are a whole host of resources that you can use. So predicting allele frequencies is what we've just been practicing. And as you click through begin, I can't on this because I haven't got the link here. I'm just showing you what the screen looks like. Okay, you can look at variation again if you click this and you can start. You can click this one, begin that. Predicting allele frequencies. And as you go through and click begin and next, there are options to download practice questions, and there are also the mark schemes with the working out. So if you feel like you need a little bit more practice with this equation, then please visit this website, okay? WJC Blended Learning, Biology, A-Level. Go to Predicting Allele Frequencies and you'll get a lot more practice there. I hope that that's been useful for you. Thank you very much for joining. Bye.